Plasmids are circular double-stranded DNA molecules that exist naturally in different bacterial species. They are extrachromosomal genetic elements that replicate independently of the bacterial genome.
Their main characteristics are moderate to small size, which facilitate their isolation and manipulation, circular shape, allowing the DNA to be more stable when removed, variable number of copies, being specific for every cell and depending on the plasmid size, and they have selection markers such as antibiotic resistance, a multiple cloning site for and a specific nucleotide sequence for ORI. There are different characteristics that a microorganism can obtain when acquiring a plasmid. One of the most common is resistant to a certain antibiotic. Also, the plasmid could be high copy or low copy. Plasmid can also provide their microorganism with virulence, and they can also become resistant to heavy metals. Also, they could be responsible of making microorganisms able to survive in presence of alternative and non-common carbon sources. They can also get the ability of creating symbiosis and acquire resistance to a certain bacteriophage by getting a new plasmid. They can also gain characteristics that allow them to colonize certain tissues and also forming tumor implants. Getting a new plasmid can allow the microorganism to change color in presence of some reagents like UV light.
Several companies sell kits for isolating plasmid DNA and basically consists on an alkalinization followed by a centrifugation to binding DNA, washing, eloting and precipitating. Finally, we can obtain the plasmid. The methodologies for plasmid extraction are different than the ones for genomic DNA or RNA extraction. In genomic DNA extraction, cells must be lysed to expose the DNA and the cell membrane should be removed using an appropriate detergent. In this protocol, proteins and RNA can be removed using RNases, proteases and phenylchloroform isoamyl alcohol which denatures proteins. By using the solution, proteins, lipids, carbohydrates and cell debris are removed through the extraction of the aqueous phase from the organic mixture. The aqueous phase contains DNA that can be precipitated with ethanol. On the other hand, RNA is an unstable molecule and has a very short half-life. Once extracted from the cell or tissues, strong denaturants have always been used in the intact RNA isolation to inhibit endogenous RNases. RNA extraction relies on good laboratory technique and RNA-free technique. The most common isolation methods can be divided into classes: utilization of 4-molar guanidinium thiocyanate and utilization of phenol and SDS. Plasmid extraction by alkaline lysis consists on 1. Cell growth and harvesting. When sufficient growth has been achieved, the cells are pellet by centrifugation to remove them from the growth medium. 2. Resuspension. The pellet is then resuspended in a solution containing trees, EDTA, glucose, and RNases. 3. Lysis. The lysis buffer contains sodium hydroxide that helps to break down the cell wall, and SDH which solubilize the cell membrane. 4. Neutralization. Addition of potassium acetate decreases the alkalinity of the mixture, so the hydrogen binding between the bases of the single-stranded DNA can be reestablished so the sdDNA can renature to dsDNA. 5. Clean up the solution and concentrate the plasmid DNA. Lysozyme is used for lysing bacterial cells by hydrolyzing the peptidoglycan present in the cell walls. This method is based on the DNA and proteins denaturalization with sodium hydroxide and neutralization with glacial acetic acid and the addition of a concentrate salt solution.
Agarose gels, there are three possible isoforms: relaxed circular, supercoiled, and linear. The difference between the bands is caused by the volume of the isoform and their migration velocity. The supercoiled molecules will move easily through the gel, so it will be at the bottom. The linear isoform will normally advance faster than the relaxed circular one, so it will be seen in the middle. The agarose gel presents some issues. The 1 kb marker from oxygen was very low definition, meaning that it was not correctly loaded or it was moved during the process. On the other hand, there is little contamination from proteins that can be seen in the chamber. There is a great amount of nucleic acid contamination, mostly RNA. It is not possible to identify the different isoforms of the plasmid since there is only one band visible and it's blurry. The bands are not equally defined, meaning that they don't contain the same amount of DNA. It is recommended to quantify the absorbance in the spectrophotometer due to sometimes shortage. This step was not possible.
The alkaline lysis method is based on the differential denaturalization of genomic and plasmidic DNA in order to separate them. In this method, the bacteria are suspended in a solution containing glucose, tris, and EDTA, which prevents DNases from damaging the plasmid while glucose helps maintain the osmotic pressure. The GTE solution can be complemented with lysozyme to increase the DNA yield. Afterwards, a lysis buffer that contains sodium hydroxide and SDS is added, which helps to break down the cell wall and denature the DNA by the high pH. The following step is the neutralization step involving a high salt solution that returns the mixture to a neutral pH and allows the hydrogen bonding between the nitrogenous bases of DNA. Because of the relatively small size of the plasmid DNA compared to the genomic DNA, only the plasmids will reanneal and stay solubilized. The genomic DNA and proteins precipitate in a complex formed with potassium and SDS, which can be removed by centrifugation. Finally, the plasmid is concentrated by an ethanol precipitation. In an electrophoresis gel, the several isoforms of the plasmid migrate and separate according to their size and shape, so 2 or 3 different bands must be observed, which correspond to the relaxed circular, the complete linear, and the supercoiled isoform in this order. In this experiment, the 3 isoforms of the plasmid cannot be appreciated as a single band that can be observed has an approximate size between 1500 and 2000 base pairs. The double voltage and running time 100 volts per 30 minutes and 120 volts per 15 minutes that were made in order to adjust the times to the class affected the migration and the definition of the bands. There is also a great amount of nucleic acid contaminations, mostly of RNA, due to the lack of treatment with RNases and its further phenolic extraction. Finally,
the most important steps during an alkaline lysis are the lysis step, which uses a buffer that contains NaOH and SDS to denaturalize DNA, and the neutralization step that involves high salt solution that renews only plasmid DNA. When extracting plasmid DNA, it is important to observe the integrity and purity by electrophoresis. 2 or 3 plasmid isoforms are generally observed: the supercold isoform, the linear isoform, and the open circular isoform. In this experiment, no isoforms can be observed in the electrophoresis gel